Our bodies have a remarkable defense system called the immune system. It protects us from harmful invaders like bacteria, viruses, and parasites. Adaptive immunity is a key part of this system, providing specialized defense against specific pathogens. Unlike innate immunity, which offers immediate but general protection, adaptive immunity develops over time and is tailored to each threat. This makes it incredibly effective at fighting off infections our bodies have encountered before. Adaptive immunity relies on two types of white blood cells called lymphocytes, B cells, and T cells. These cells work together to recognize and eliminate specific threats. When our bodies encounter a new pathogen, it takes time for the adaptive immune response to develop. During this time, we may experience symptoms of illness. However, once our adaptive immunity has learned to recognize a specific pathogen, it can respond much faster and more effectively the next time we encounter it. This is why vaccines are so important. They introduce our bodies to weakened or inactive forms of pathogens, allowing our adaptive immune system to develop immunity without causing illness. The adaptive immune response is characterized by its specificity and memory. Specificity refers to the ability of lymphocytes to recognize and target unique molecules called antigens on the surface of pathogens. Memory means that our immune system can remember previous encounters with pathogens, allowing for a faster and stronger response upon subsequent exposures. This remarkable ability of the adaptive immune system to adapt and remember makes it a cornerstone of our body's defense mechanisms. The effectiveness and complexity of the adaptive immune response highlight the intricate workings of our bodies. Understanding the principles of adaptive immunity is essential for appreciating how our bodies fight infections and how medical interventions, such as vaccines, can bolster our defenses against a wide range of pathogens. B cells are a type of lymphocyte that plays a crucial role in humoral immunity, a branch of adaptive immunity that focuses on fighting pathogens found in body fluids like blood and lymph. These cells are produced in the bone marrow and mature in different parts of the lymphatic system, such as the spleen and lymph nodes. Each B cell carries a unique B cell receptor, BCR, on its surface that can bind to a specific antigen. When a BCR encounters its matching antigen, the B cell becomes activated. Upon activation, B cells undergo a process called clonal expansion, where they rapidly divide and differentiate into two types of cells, plasma cells and memory B cells. Plasma cells are short-lived but highly efficient antibody factories. They secrete vast quantities of antibodies, which are specialized proteins designed to bind to the specific antigen that initially triggered the B cell activation. These antibodies act like guided missiles, targeting the pathogen for destruction by other immune cells or neutralizing its ability to infect cells. Antibodies, also called immunoglobulins, IG, are proteins that play a critical role in humoral immunity. They are Y-shaped molecules with two identical antigen-binding sites at the tips of the Y. This structure allows antibodies to bind to antigens with high specificity, much like a key fitting into a lock. There are 5 main classes of antibodies: IgM, IgG, IgA, IgE, and IgD. Each class has a unique structure and function. For example, IgM is the first antibody produced during an immune response, while IgG is the most abundant antibody in the blood and provides long-term immunity. The interaction between an antibody and its specific antigen is a critical step in the humoral immune response. This interaction, known as the antigen-antibody reaction, leads to the formation of antigen-antibody complexes. These complexes effectively neutralize the pathogen by blocking its ability to bind to and infect host cells. Additionally, the formation of these complexes enhances the recognition and destruction of pathogens by other immune cells such as macrophages and neutrophils through a process called opsonization. T cells, another type of lymphocyte, are the key players in cell-mediated immunity, which focuses on eliminating intracellular pathogens, such as viruses and some bacteria that have infected our cells. T cells originate in the bone marrow but mature in the thymus, a small gland located in the chest. Similar to B cells, each T cell possesses a unique T cell receptor, TCR, that recognizes specific antigens. However, TCRs cannot recognize antigens directly. They require the help of specialized molecules called major histocompatibility complex, MHC proteins, which are present on the surface of other cells. There are two main types of T cells: helper T cells, TE cells, and cytotoxic T cells, TC cells. T cells play a crucial role in orchestrating the immune response. When activated by recognizing an antigen presented by an antigen-presenting cell, APC, such as a macrophage or dendritic cell, these cells release signaling molecules called cytokines. These cytokines stimulate the activity of other immune cells, including B cells, TC cells, and macrophages, enhancing their ability to fight the infection. TC cells, also known as killer T cells, are responsible for directly eliminating infected cells. When a TC cell encounters an infected cell displaying its target antigen on MHC class I molecules, it binds to the infected cell and releases cytotoxic molecules, such as granzyme and perforin, which induce apoptosis or programmed cell death in the target cell. This targeted killing of infected cells prevents the spread of the pathogen and limits further damage to the body. The coordinated action of both B cells and T cells is essential for a robust and effective adaptive immune response. While B cells target and neutralize pathogens in body fluids, T cells eliminate infected cells and orchestrate the overall immune response. This intricate collaboration between different cell types ensures the efficient clearance of pathogens and the resolution of infections. The adaptive immune system is a complex and powerful defense mechanism, but it is not without its flaws. Sometimes the immune system can malfunction, leading to a variety of disorders. These disorders can be broadly classified into three categories: immunodeficiencies, autoimmune diseases, and hypersensitivity reactions. Immunodeficiencies occur when the immune system is weakened or absent, making individuals more susceptible to infections. Autoimmune diseases arise when the immune system mistakenly attacks the body's own cells and tissues, leading to chronic inflammation and damage. Hypersensitivity reactions occur when the immune system overreacts to harmless substances, such as pollen or pet dander, causing allergic reactions. Understanding the complexities of adaptive immunity is crucial for developing effective treatments for these disorders. Researchers are constantly working to develop new therapies that can modulate the immune response, either boosting it in cases of immunodeficiency or suppressing it in cases of autoimmunity and hypersensitivity. These advancements in our understanding of the immune system offer hope for developing more targeted and effective treatments for a wide range of immune-mediated diseases. In conclusion, adaptive immunity is a remarkable and intricate system that plays a vital role in protecting us from a constantly evolving world of pathogens. The ability of B cells and T cells to recognize and target specific antigens, coupled with the immune system's capacity for memory, makes adaptive immunity a cornerstone of our body's defenses. While the immune system can sometimes malfunction, leading to disorders, ongoing research and advancements in immunology offer promising avenues for developing novel treatments and improving human health.